it's not the worst day to get the food here. Okay, so what we are looking right now is what we call the Ethiopian collection that we have here. And the reason that we have this material here, just to have an idea, here we have a picture illustrating the origin of the modern cultivars of coffee Arabica. So basically, if you get the white coffee, right, it was about 600 before Christ, so 75, right? And this material was taken to the Yemen, and after that, I have a split that some material was to the island of Java and the island of La Réunion. And after that, we have materials that went to Botanic Garden of Amsterdam. And this material originated the materials that we call typica. And the part of the La Réunion island originate the material that we call Bourbon. And the majority of the modern cultivar that we have is a cross between Típica and Bourbon, from Mundo Novo and Catuai. That is basically 99% of material that is planted in Brazil, that's cultivated in Brazil, is Catuai and Mundo Novo. And you think about genetic diversity of this material. You come from a broad genetic base, and you have really a kind of constraint in terms of genetic diversity here. So these are huge problems for tomorrow and all the breeders that try to work with new characteristics in terms of agronomic characteristics for coffee. So what we have here, so we have this huge collection from, not huge, but we have 130 materials that we call like white type coffee, right? So they came from Ethiopia. This was a mission from FAO in the 60s that bring material from Ethiopia and rescue some materials over there, since you have like several problems in terms of well, political problems that will damage the conservation of coffee over there. So they rescued this material, and there is a huge collection in Cartier, Costa Rica, and there is a part of this collection is in IAC, in Campinas, and you have part of the collection here, right? So we start to explore this collection in terms of some agronomic characteristics, some biochemistry characteristics of the coffee, for sugar content, for diterpenes, oil, and we saw that there's a lot of variability from the material that we collected here. So basically what we have in this part here is the old collection. The collection a few years ago was in really bad shape, so we start to improve in collaboration with people from Sirhat. We improve the collection that we have here, and also we start to do a new collection of those material here. So we have replanting the S1, the seeds of this material here over there. So that's the reason that we have these big trees here and these small ones here that we just start to renew this collection here to continue to do all this kind of analysis that we have here. And we have a lot of genetic diversity here for shape of the plant, for maturation time, for flowering time, and also from characteristics that are related with quality. So, basically, caffeine, right? I don't know if you remember, some time ago in Nature, there is an article about a coffee tree that was caffeine free, right? And basically, this plant came from this collection. Not this collection here, but the collection at the ESC. They have this plant that it's caffeine free. So they came from the Ethiopia. It's the same collection that they have there, we have here. So,
Laurina.
Como
é que chama?
Não, não é laurina. It's another material that is a white type material that came from the Ethiopia. They have a mutation for the gene that is responsible for caffeine. It's a natural mutation that they found in the field. We also have some variability about caffeine here in this material, but I think we didn't find anyone that's caffeine free. We found some very low levels Very similar to the ones that they have in EC, but not as low as they have there. It's similar but not as low, but you can use us for low caffeine breeder if anybody likes low. You know, markets sometimes like, I don't like, I like my coffee tons of caffeine. And this is material you can so we try to analyze this material and give information after that to tomorrow to improve the coffee and in terms more specific for some characteristics, agronomic characteristics that he thinks that will be important.
We are conducting F1 plants, crossing together here of Cirrade, né? Low content caffeine plant, né? With conventional cultivars, and the other is crossing between trichinosis resistant plant with small leaves to improve the photosynthetic system and less diseases, and it is easy to apply fungicides, chemical products to protect against the disease and the infections. This is one part of the research that is consequence result of this research informed by Felipe.
Yeah. So basically that's what Timur said, like we start to find some nice characteristics of the material, and then here tomorrow then we can do cross and make populations, and then start to do breeding from this material. So that's basically what we are doing here. And so we have not only this material that I said, and the material that tomorrow said, that the cross also they have over there for the population that we call mapping population, because we like to do the breeding and also try to do genetic mapping of this material as well. Yeah, it's for we also tried a little bit for drought. We have some work in terms of drought tolerance. So here you see that there is one part of the collection that is they have just black tubes that for irrigation, and over there is another part of the collection that is no irrigation at all, just to see if you can, in a year that you have problems with drought, we can see the difference here. So we try to look for something about that too, in terms of the collection, what material is to be more resistant, tolerant to drought.
Taste the coffees?
Not yet. I think Brigida maybe start to do some work on that.
The thing is,
but this is not.
There is a big variability here, and we don't have often enough coffee to make to have reliable proof of tasting. And that's why having more plants here, we will be able to have quantities that will be sufficient for it. I have to add with to what Philippe said, that the genetic analysis is you visited the lab the other day, is parallel to that agronomical studies, because we are trying to relate the genetic analysis with the characteristic we observe in the field, and this through what is called mapping. There are two ways: getting sufficient data in the field. Also for drought we have trials in controlled conditions, and we have also some replication of this in a place where there is a real drought of 6 months close to Brasilia, so that we will gather data from various conditions and compare with the genetic data in order to be able to locate genes or parts of the chromosome which are related to some characteristic, and so the breeding work of the breeder will be much faster, and this work gives results.
This is a good point. Like I forgot, like everything that I mentioned in the lab, it's a lot of it's related to the work that Pierre is doing here in the field, and we try to connect with that and see if we can get something.
You have a good illustration here of what you can see here as well is the big differences between the trees, and this is a representation of what you can find in Ethiopia. The varieties are quite different.
Now is evaluating oil, protein, caffeine, sugar. Depends.
It depends on oil, depends on chlorogenic acids we evaluate as well.
Vitamin B. É aquele outro composto, né, relacionado. Ok?
Ok?